Welcome to Barry's in the Interim podcast, where we explore the cutting edge of innovative clinical trial design for the pharmaceutical and medical industries, and so much more. Let's dive in.
Welcome, everybody, back to In the Interim. I am your co-host today, Scott Barry, and I'm joined by my common co-host, Kert Viele, here, and we are here to talk about something, and Kurt will occasionally say, "Oh, I've got a good topic for the podcast," and usually it's something that's beneath the craw a little bit, maybe within the pet peeve, but I think a really important topic, a really important misunderstood topic, and that is bias in early stopping in clinical trials. So, welcome back to In the Interim, Kurt.
Thank you.
All right. So we are in the interim, and we're talking about bias of interims, sort of an interesting aspect of this. I thought I'd introduce the topic by telling a bit of an old story. And this happened relatively early. So I started designing trials in 2000. Before that, I did a little bit of clinical design, but this was early in Barry Consultants, and was working with a medical device company. And they had an estimate of their effect size, but thought that they could be quite a bit better than that. So an adaptive sample size with early stopping for superiority made a lot of sense for the trial. So fairly standard 2 interim analyses for early success, could go to the final analysis, medical device. Nothing controversial in the trial design. We can control type 1 error. It's not the issue. Pretty straightforward. And so we were working on the design, and then I got word from the company that there's a problem with stopping. And the company was associated with a group that was going to run the trial, and they had an issue with potentially stopping the trial. I thought, okay, that's odd. Let's talk to them about it. So the concern was the bias in the estimate that comes out of the trial, and it's important for medical decision making to have good estimates of the effect. And the concern was that the early stopping biases that estimate. And so, okay, that's great. And one of the wonderful things about simulation is we can simulate the bias. So it was a response outcome, success and failure. And I simulated the trial under a number of potential true effects and calculated the bias of the trial. And it was really relatively small. It was approximately 1% in a rate of success of, we're looking for 30, 40, 50% success rates, and it was a bias of about 1%, which I thought was a minor thing relative to the benefits of sample size, patients' time to get a success should the effect size be that large. And the individual I was talking to, and I was actually talking to 2 people, one senior member, and I'll tell you who the senior member was in a bit, but at the time I had no idea either of these people, and one was a statistician. It turns out the statistician doesn't say anything during this conversation. And the person says, "Oh, we're seeing biases larger than that." And asks, what is the bias if you restrict to trials that hit the early success? And I said, well, that's not bias. Bias is when you look at all the trials, what is the average effect relative to the truth? If you restrict it to trials that hurt that early success, that in and of itself is a bias. Surely you're going to get much larger effect because you have to have a large effect to win. And showed what that was, but that's not bias. He says, well, I don't actually know what the mathematical definition of bias is, but that seems really problematic to me. And it was sort of strange in that. I said, well, if you look at any trial, if you look at a fixed trial and look at only those that are successful, you're going to see much higher estimates. Claiming success, it has the same sort of bias. What was fascinating about it was it got the advice, and the group running the trial was very closely associated with the device company and needed the approval of the group. Turns out they had to get rid of all the early successes. It was very, this is really strange, in the whole thing. So the individual that I was talking to, I met for the first time, was Gordon Guyatt. And we're going to come back to that story. But Kert, this is a thing that I think a number of people know that this is a potential issue, the bias, and think it's a big issue with potentially stopping early.
So I think, one of the key things you brought out about that is, what is the definition of bias? What are you worried about? And, ordinarily when we take this in a class in undergrad or so on, we talk about all trials. Everything that could happen, average them. On average, you get the right answer, the right response rate. Whenever you cherry pick, you're going to generate some kind of bias because you're cherry picking. I do have a little more sympathy, I think, maybe than you do on this, that if I know that I stopped at the first interim, I stopped at half the planned sample size, I know these are the situations where I might see the high part of the distribution. Is that a concern, and how big of a concern is it? I certainly can see people wanting to compute that. So this is going to be a key part of our story going forward.
Yeah. So we'll come back to that. And there's no question that trials that show statistical significance overestimate the treatment effect. I think we all understand that. In it, a trial that stops early is going to provide likely an overestimate. In it. It all depends on what you think the potential range of effects are. But let's come back to the definition. I think that this is so important, the definition of bias in it. So what we think of it is, what's a biased estimator? An unbiased estimator would be one that, when you run your experiment and you have an estimate of what it is, whatever it is you're trying to estimate, the success rate of a treatment, if on average that estimate is the real answer, we call that unbiased. So the expected value of the estimator we're using within our experiment is equal to the estimand we're trying to estimate. We call that unbiased.
And so we're thinking about everything that could happen. I mean, this is all, if I do an interim, I don't just have early stopping for success, I've got early stopping for futility. So I'm averaging, I stopped at 50 for success, I stopped at 50 for futility, stopped at 75 success and futility, went to the end, stopped for success or futility. All of those things average together. If those all average out to the right answer, we're unbiased. And now we're going to get into, do we need to look at each of those 6 things separately?
Yep. Okay. So, and we can calculate that if you do early superiority, what happens is, within the trials you could run, those trials that hit early superiority are depending on the real parameter, the real truth, likely is overperforming.
Yep.
Within that. And you stop at the point it's overperforming, and it doesn't go the rest of the trial and regress towards the parameter. So when those trials stop and you report that, we know that's biased. By the way, my pet peeve, and I know we generally have one of Kert's pet peeves, but one of my pet peeves is, bias means you can't use it. that all of a sudden it's done. Bias is okay. We can adjust for it. We understand it. We want to know, what is the size of the bias in the circumstance. It's just, bias doesn't mean you can't do it. And I think there's bias, you can't do it. So we understand there's bias. So let's talk a little bit about that, Kert. Within a toy example, let's talk about, what is the size of this bias?
Well, and so let's start back up a little bit further, and let's start for a fixed trial, just a straightforward, I run a trial, and my toy example here is I just have straight normal data. I'm estimating a mean. I've arranged a trial, sample size is 100. I'm going to run a z-test at the end. Everything is as basic as it could be.
One randomized experimental to placebo.
Yep. Standardized effect size of 1 here, just to make things easy.
Yep.
Okay. So if I take all trials and the truth is 1, so I come in with what I powered for. The truth's 1, I look at all trials, I get an average of 1. It's unbiased. They're the same.
No futility, no superiority, 100 patients, read it out. Yep.
Okay, so now all of those trials that were unbiased as a whole, I can split them. I can talk about the ones that won, p less than 0.025, and the ones that lost. So if I look at the ones that won, they have an average of about 1.06. So they're biased high. They're 1.06. They're compensated for by the ones that lost. And so you have to have this compensation going on. In fact, if you make the p-value, the stronger the evidence, if you make the p-value less than 0.0001, for example, so you're like, oh, look, I won, this is great, I have a great drug, and so on, somebody could come back and go, oh, wait, if you focus on the trials with p less than 0.0001, their average effect is 1.34. It's 34% bias compared to the truth of 1. So you want to be careful to go, I can't accept anything that's biased, because you're essentially saying the stronger the evidence, the less I believe it or something.
Okay, so under the truth that the real effect is 1, where your trial is 90% powered for 1, so most of the trials win. Only 10% of the trials don't hit 0.05. If you restrict a calculation of what's the average effect for those that win, you get 1.06. So there's a 6% bias just in successful trials in that scenario. Now, if you were to do this under the null, the bias would be enormous for successful trials.
Yeah, you need about 0.6 to win. Usually a 90% powered trial, you need about 60% of the powered effect. So your bias has to be at least 0.6 in those cases under the null.
So bias depends on the truth of the effect. If the effect is 3, you're unbiased because every trial wins and there's no bias to that. So so we're addressing powered for the effect of 1 within that scenario.
And if you go the other way, sorry to interrupt you, but if you go the other way, if the true effect was 1.5 or 2, it's higher, then you have 100% power and you get no bias whatsoever. It definitely depends on the true effect.
Yep. What happens if I add futility?
So then you're going to end up where, now, of course, futility in a fixed trial, you just have the winners and losers. But if we start to add into the group sequential, we add futility, that's going to be a compensating mechanism for the bias, or the upward bias. Okay. Downward bias.
But we have, I forgot, we've done fixed trials. All we did was restrict, and you have this 6% bias of successful trials.
Yep.
Now, what if I do, so you added into this O'Brien-Fleming stopping at 50 and 75.
Yep. And so if I do that, and I don't have any futility. So futility is important here because it does compensate. Then I'm going to end up overall, I get an effect about 1.15 as opposed to 1, so 15% up. So that's certainly meaningful. You got to weigh that against the fact that I'm stopping early to help patients, and I don't know the effect is 1. It might actually really be good. If the true effect is 1, and I stop at 50 in that, half the same.
Let's back up. Make sure there's this. So over all possible trials you run,
1.15.
Some of them stop at 50, some stop at 75, some stop at 100. Not just successful trials, but all trials.
All trials.
All trials. The average.
Sorry, all trials is 1.07. So thank you for correcting me.
Okay. So 1.07 is that. So the bias in a group sequential O'Brien-Fleming design, there's a 7% bias.
Yes, with no futility.
Yep. So the question is, if we just look at that, that's the bias of adding it into the trial within that scenario. Now,
which isn't different than the fixed trial that we just had. That was at 6.
But that was restricting to only successful trials. Yep. So if FDA says you're running this group sequential design, what is the bias of adding in those early analyses? Assuming that your effect size is 1, it would be 1.07. There would be a 7% difference from the truth of that in that. Now you can adjust the estimate for that. There are statistical ways to do that within that. But even that bias, now we are able to correctly claim superiority, perhaps halfway through the experiment, 75% of the way through the experiment. The savings in patience and times to get the right answer at the cost of this 7% bias, that's the sort of question. And usually that seems like a no-brainer trade-off to me within that, in those scenarios for that. Okay, but now what happens if you only look at trials that win in a group sequential?
So only the ones that won, you get 1.15. So it's going to be a little bit higher because you don't have the losers at the end.
And that includes the probability you win at 50, at 75, or you go to the end and you're successful at the end.
Yep.
Within that. And so there is that increase. Winning trials in a fixed trials were 1.06. Now it's 1.15 within that. Now, what if I only look at trials that win at 50?
Now, that one's big. That's 1.5.
1.5. Yep. Now, that's not bias, right?
It's not biased by the definition. I have a little more sympathy and kind of a little uncomfortable.
Okay. But so that in that scenario, and of course, in the scenario where you need to see that larger effect size, on average 1.5, to win that, to be statistically significant in that scenario. If you only look at those scenarios, you get biased because the truth is 1 and you have to have a value bigger than that. Now, if the truth is 2, that bias is going to be basically almost zero.
Basically zero,
yeah. So one of the points I think you would make is if you knew the effect was 1, you wouldn't do an interim at 50, in that scenario. Or if it's incredibly unlikely that the scenario's as big as 1.5, you wouldn't do that interim.
Yeah, we always, when we show this to clients or when we do this, we back solve what effect do you need to stop for success and futility at each interim. And we often ask clients directly, especially on the futility side, are you comfortable stopping the trial here? Would you be willing to basically give up at this point? That applies for success. Would you believe this result? If the answer is no, then I have a lot of concerns about doing the interim. Again, unless there's some public health, thing we need to have this answer as quickly as possible. I still trust the drug works in that case, but I do worry that maybe it doesn't work that well.
Yeah, and so the estimate of that. So a very common situation, and the situation I was in the device trial is that the company might believe in the 1.5 is right, but they want to make sure their trial is powered for 1 because it's clinically still a valuable device. It's good for patients. And so they power the trial at 90% at the maximum for the 1, but they really think their device could be as good as 1.5, 1.75, or 2 in that scenario. And then the bias under those scenarios is much smaller, and it's a much more reasonable thing to have a variable sample size. They actually think their effect is 1.5, and they want to run a trial that's only 70, but they go to 100 potentially to still hit a clinically meaningful effect within that scenario. And then you're talking about biases of 7% by doing this, even under the 1. It's much smaller in the 1.5 scenario.
And I think this is where I really put on my Bayesian hat, so to speak, even though I'm computing biases. When I see that data, it's stopped at 50. I've got an effect size around 1.5. All of these bias calculations assume that you know the truth. When I get the data, I don't know the truth. I got to figure it out. And so if I knew in my head it can't be bigger than 1, then of course I don't trust the 1.5. But if I've walked into the trial where this might not work at all, it might be 1, it might be 2, it could be any of those, then I start thinking to myself, okay, if it were a null, it hardly would ever stop. So I don't see many of these. I know they're biased, but they just don't happen very often. They happen very rarely. If it's 1, it still doesn't happen all that often. I don't stop at 50 a ton. If it's 2, I stop at 50 all the time. So when I see that 1.5, it's more likely to be one of the 2s and is the right answer than it's one of the biased ones. It's more likely to be an unbiased truth than one of the biased falsehoods. And that gives me a lot of comfort.
Yeah. You'd probably make a small adjustment, like 7%, and think it's probably not quite, 1.5 of what was observed under that scenario.
I'd adjust by my prior. There's a clean Bayesian answer to this,
Yes.
which we've had our frequentist hat on, but every Bayesian would just compute the posterior and it works. It corrects the bias automatically.
Yep. Okay, so we can calculate the bias for that. And as we do more complicated designs, and you could add response adaptive randomization, which we've done a recent episode of. We could add arm dropping in here. We could add patient changing. We can do all this. We can calculate the exact bias under a number of scenarios. One of the really nice things about simulation. So we can calculate bias and we know what it is. Mathematically, we can calculate it.
And we should certainly emphasize, this is one of the things that in the regulatory world, we've been doing a lot more. The last 5, 6, 7 years, regulators have been paying a lot more attention to this, asking for it. And so we've been providing these, and for the most part, things are going fine, but we have found a few examples and we've changed the designs in response.
And the FDA draft guidance, the FDA guidance on adaptive designs, the ICH 20 adaptive designs, that's draft, they all mention calculating the bias in it. I don't know if we have submitted a design over 27 years of doing this to the agency where they've had an issue with the bias.
That sounds fair to me.
I think, yes, if we were to do a sample, if we were to do an interim at 10 patients in your 100 patients example, there could be an issue with that.
You're cherry picking because we don't actually let it get to this point.
Right. So we wouldn't submit that design when we calculate the bias. We say, wow, if I were a regulator, I'd have a problem with that sort of thing. So, it's something that they understand, they recognize when they have to write an FDA label. I think there's comfort there with it, in it, and we can calculate that exactly. Okay, so I keep saying that over and over again, and people are, why does he keep saying that? We can calculate that sort of thing. Coming back to my story.
I think this is definitely your pet peeve episode.
Yeah. Okay. All right. I'll take that. But you brought it on. You said, "Hey, we should do an episode." Oh, and I remember this story.
Triggering Scott is a hobby of mine.
Yes. So Gordon Guyatt, you may not know Gordon Guyatt. What happened is, within a few months of that interaction, a paper came out in JAMA, and the lead author is Bassler, and this is Bassler et al. The senior author in it is Gordon Guyatt, and it's on behalf of something called the Stop It Study Group, Stop It 2 Study Group. And the paper is, so this is, this comes out in March of 2010 and in JAMA, and it is stopping randomized trials early for benefit and estimation of treatment effects, a systematic review and meta-regression analysis. So I'll read you the conclusion of the paper and then describe a little bit about what they do. And by the way, I will say this as I'm describing this, that in this many years, I think this paper is just wrong. I think it's misleading. I think JAMA should have never published it. But we'll come more to that, but just to sort of set the scenes for it. So the conclusion is truncated randomized clinical trials were associated with greater effect sizes than randomized clinical trials that's not stopped early. That's the conclusion within this. But one of the sentences in the paper says, statistical modeling suggests that randomized clinical trials stopped early for benefit, and they call these truncated randomized clinical trials, will systematically overestimate treatment effects. And empirical data demonstrate that truncated randomized clinical trials often show implausibly large treatment effects. So a lot of people reading this paper, looking at it, come away with, you shouldn't do interim analyses. It creates implausibly large bias. This is bad. The name of their group is Stop It. And I don't think they mean trials. I think they mean it. And he literally, in that example, prevented a company from doing a group sequential design because of this issue. Okay, so what did they do? And their goal was understanding we can mathematically calculate bias, but what do we actually see out there for bias? So they went out and they found, through a search, they found trials that were stopped early, truncated early for superiority. And you can go to their consort diagram. They originally found 195 trials that were stopped early. Then they looked for trials of the same question, other randomized trials of the same question. And they were going to compare the effect of these other trials to the ones that were truncated early. And by the way, in their search of where they had 195 trials stopped early, and then they looked for other trials, they found some more that were stopped early. They moved those over to this, calling them truncated. And
if you replicated the result, you moved it back over and it didn't count as a replication.
Well, if you got early truncated,
yes, but if you repeated the experiment, you didn't get credit for the replication of it because you truncated it early.
Yep. You go into, they're going to create largely a treatment group of trials, which were ones that were stopped early. And then the other group are the same question that were not stopped early.
And some of them could have been.
Some of them could have been. Some of them were statistically significantly successful. Some were not.
And it's worth mentioning that if you could stop early and you don't, those trials are biased low. So we mathematically know they're going to be a low group.
So and this was their empirically what happens out there. So they took this set of trials that were stopped early, And then they compared to the same question that were not stopped early. And they found that the difference was about a 30% difference in the estimate of the effect. They did a relative risk and it was 71%. So a 29% relative difference in the effect of those stopped early versus those that were not. And they jump up and down and say, this is enormous. A 30% effect. So in your particular example, we had that effect of 1. If we did something that created an average effect of, say, 1.3, we might be concerned that that experiment provides bias. And that was their experiment that they did in this comparison, and they published the results of that.
And we have that result. So if you look in the example we gave, the 100 sample size and so on, if you look at the trials that went to the end and won, their average effect is only 0.87 compared to 1. They're biased low by a fair amount, 13%.
Yep. So in a way, they present this result, and think you're all clinical trial people, and you understand that we in a clinical trial, as simple as 2-arm trial, we have a treatment group and a control group. And we randomize people into those groups and we compare their results. They took this group that the trial was truncated early and they call that group 1. And then they take trials that were not stopped early. If it was stopped early, they moved it over into group 1. That were not stopped early and they called that group 2. And they said, wow, group 1's got a better treatment effect than group 2. But by the way, if you ran a clinical trial like this, it would be that you run a trial and you have patients, and the ones that respond well, you put them in treatment 1. And if they don't respond well, you put them in treatment 2. And you say, wow, treatment 1 is better than treatment 2. That would never get into JAMA, but somehow this got into JAMA. Interestingly, when this was published, there were many letters to the editor. I was a part of one of them with Brad Carlin and Jason Connor, where we point out that this is faulty and wrong. But the names of the people that wrote in saying this was wrong was somewhat impressive. Another one was Steve Goodman, Don Barry. My father wrote a letter to the editor, a different one than the one I was on. And Janet Wittes were on that one. Another one of these letters was Ed Korn and Margaret Mooney. Another one was Susan Ellenberg, Dave Demetz, and Tom Fleming, also pointing out the faults of this. And so there were a lot of people that pointed out that this was a bad study in it, but yet still and by the way, think about it, that you go in and you take trials early. If they would have done the same thing where they went out and found trials that were statistically significant, and then they went out and found trials that any other trials run, even some that were statistically significant, they would see that that's biased within that. And that's largely the bias that they saw in that. And we know there's a bias to stopping early. We can calculate it mathematically. So when their conclusion was this is implausibly large, I think they understood the mathematical calculation of 7%, and we're seeing 30%. They even study things like, did the DSMB have some investment in it? All the looking for chicanery here or something like that within this. And it's like, no, this had to happen. If you ran an experiment where you took the biased sampling that they did, you would see this 30% bias. This is nothing.
I'm laughing, Scott, because I was truly successful about getting you riled up today.
Yeah. So this paper's sort of number one on my list. You know others. We'll do other podcasts of other ones within the scenario. But and it was kind of amazing at the time because I tried with fault to explain this to Dr. Guyett. In that I was unsuccessful. The paper came out, and I think there was nobody that sort of said this is odd. He did a debate with Steve Goodman after this paper came out where Goodman explained this. And I think he largely, I'm not sure he sort of understood the intricacies, and largely it was like their rebuttal to these responses that, well, boy, look, this is big. And yeah, by definition, you bias sampled in a way that created this. Nobody should be surprised by that.
I mean, make sure we summarize this properly. We certainly are claiming there's bias. So there is some bias. We can compute what it is. That has to be balanced versus the advantages. We stop early for reasons. We want to protect patients. The Dawn trial, for example. This is a trial that thrombectomy, great effect, interim at 200 patients, maximal sample size, I think 500. Am I remembering that right? So,
stopped at 200.
Stopped at 200. So enormous effect that's panned out in practice. This is the standard of care now. If that trial doesn't stop at 200, it is years before this. There are other trials with thrombectomy. I don't want to claim it's years before that reaches the market, but it's years before that evidence is actually in front of people.
Yeah, and so there's huge benefits, and also, by the way, it allows you to have a wider range, to have a higher powered trial where you wouldn't run that single maximum because you think it's wasted because you don't need that many. There's a huge benefit to having a flexible sample size, and this painting it's biased, you shouldn't do it, ignores all of these important factors to trial design.
And I do think you have to incorporate that entire range of evidence, or entire range of possibilities. I can't just compute bias on suppose the null is true, suppose this effect is true. What's the range? What's the bias for each of these? What's the likelihood I'm actually going to see these? It's like a doctor getting a diagnostic test. Here's what's in front of me. Is this a basically unbiased real result, or is this biased high? And if it's plausible, it's probably a reasonably unbiased real result.
Yep. All right.
You need a second to calm down?
Yeah, I'm calmed down. Calmed down. Yep,
all right.
Yep. In this one. All right. So, and I'm not lost the bit of the irony that we're talking about bias of interims, and here we are in the interim in this scenario. But that's the world we live in here. I would encourage you to go look at the Bassler paper. If you have your study groups and you read it, I think you should read it and talk about the paper, but whether you think it's reasonable. Read the comments as well. I think you'll enjoy the various people commenting.
And look at your interims when you're doing them. They're not every interim shouldn't be there. So feel free to compute these quantities and restrict to what's reasonable.
All right. Well, I appreciate you bringing this topic to me, Kert, and you see what other neat topics we have coming there. And we're going to stop this. We're going to stop this episode a little early here, Kert.
Okay.
I think that's appropriate. It's a little early, and we are biased here. And we will be biased next time. So thank you all for joining us. And until next time, we will be here in the interim.
Thanks, Scott.